Now we are going to have a discussion, a panel discussion. Of course, it will be shortened as it were because we have had so many interesting talks which lasted slightly longer than planned, but we have now the way, not yours, no, I didn't tell you, it was a general comment, thank you, Ed, and also in terms of our panel discussion. So I have sampled, there are a large number of different questions which came along, which we are definitely not able to be handling all of them. So I have sampled them and put them in a small number which are related to one another anyway. So the question is specifically for the two talks on the small molecules, the oral therapy, about off-target. So if I may get some comments by both Brian and Beth about off-target, about these small molecules.
Yeah, definitely. I'll go ahead and go first. So we've looked at RNA-seq, to really address this issue. So at levels at which we would see much higher concentrations than what we would target within the clinic, we have not seen any really aberrant off-target splicing that's occurred. So we're confident in the doses that we would go with, that we would be in a safe range for these patients.
Yep, and so we've actually done, obviously we do have one off-target, which is SMN. The gene that which led us to proceed with treating SMA patients. We actually don't think that's going to be a problem in HD patients because the SMN protein is very tightly regulated, and we only see increases in the protein in SMN deficient animals. So we don't expect that to be a problem. In terms of other targets, we actually had published a paper previously where also from RNA-seq data, we did find other off targets, but actually only one of them persisted besides Huntington after a washout period. And for that one, we only saw changes in the in RNA and not a concomitant change in protein. So with our weekly dosing, we actually don't expect to see changes in any other proteins in patients.
And related to that is a question of allele specificity, and what would happen in the reduction of wild-type Huntingtin. Would you have some comments on that? Maybe Beth first.
Yeah, I saw there were a few questions, and apologize if it didn't come across clear that this neither of these compounds are allele selective. I mean, that would be really nice to have an oral drug that was allele selective, but at this point, they are not. And I think, we are very cognizant of not wanting to overlower normal Huntingtin protein. We don't really know. We know some about animals. A lot of it is done in juvenile animals where lowering too much Huntingtin is certainly not going to be a good thing. But what we have seen in our SMA patients, and we've started as young as 6 months in these studies, or even 5 months, is we really didn't see any consequences of lowering. In that case, it'd be lowering normal Huntingtin. Of course, they have 2 good copies of the Huntingtin gene. So, perhaps it might be different in a patient that had 1 mutant protein. So I think it's something that we will keep an eye on, particularly in the periphery, where we expect to see with branaplam more lowering than in brain. And it's something we'll just have to keep an eye on and measure. We know what to look for. And we're hoping also to learn a little bit more from the Tommy Nursing data about, how much lowering they actually saw and whether some of the increase in ventricular volume was due to removal of too much Huntingtin. So I think there's still a lot to learn.
Brian?
Yeah, so it's a very similar concept. So we're looking at that with PTC518 being designed specifically for HD. We feel confident in lowering the HTT levels in a manner that is going to be safe and consistent with the predictive likelihood of having clinical benefit. We'll monitor it, obviously, and keep an eye on it. We think that what we had seen within the Roche trials, and it was ASO specific, so maybe not necessarily related to Huntingtin lowering per se, but more related to an ASO off-target effect or an ASO complication. So we're confident in what we've seen. We've looked at the literature, we've talked with the experts, looking at our research and waiting for our data to come in to ensure that we've got the right dose dialed in.
So Beth and Brian, are you working towards assays that would allow you specifically to monitor whether you're lowering Huntingtin too much? aside from the normal clinical safety measures, and that will be part of the studies anyway.
You mean specifically in peripheral tissues? Is that what you're referring to?
For instance. Mm-hmm.
Yeah. So, I mean, obviously we can measure, blood, PBMCs might be more useful. We did consider doing, other tissues, maybe, fibroblasts from skin, but, I think that, we don't really want to be overly invasive in sites where we don't need to. I think the good thing is that, with both of these compounds, they're fully reversible. So, I mean, I think there's been some animal studies, Jeff Carroll did a nice study showing, if you remove, 90% of Huntingtin from animals, it's not a good thing. You see effects in liver, I believe it was. In this case, if it happened, you stop taking the dose and it will recover. So I think we will measure liver enzymes, pancreatic enzymes, cardiac function. We measure everything. And in addition, we're looking for potential benefit, which I think is another thing to people, first and foremost, it's measuring any safety implications of overlowering. But in the long run, we're actually hoping that there could be some benefits, particularly with, muscle wasting. If we could show some slowing of that, that would be a very beneficial thing.
And I think one of the things with 518, to Bernhardt's question, and we'll move on to that. We're looking at this, so with 518 being not effluxed within the target organ, we're not anticipating as much. We'll see partial lowering in the periphery, so we're much more comfortable in that space, believing that we're not going to run into challenges and issues. But we're still finalizing what the phase 2 is going to look like. So once we have more details, I can share those later.
If we switch a little bit the topics now, there are some questions for Catherine about the Ventolol. One is about related to what we said about tetrabenazine before. Would the company think about a head-to-head comparison? This is one, and a very short question also about it doesn't cause depression, as you mentioned, the compound, but yet there is a depression scale which is being used. So would you like to comment briefly on these 2 points?
Yes,
Catherine,
briefly to point to the comparison, head-to-head comparison with tetrabenazine. Yes, for sure it will be a requirement, particularly of the European agency, but it is for the phase 3. So we for sure will have to show that we have a significant benefit against this reference drug. And regarding depression, of course, it's very early to say that our drug is much safer regarding this issue and depression, but we can say first that the drug Ventolol, which has been used in hypertension since more than 30 years, neither in its clinical development in more than 3,000 patients, and nor during its use, commercial use in more than 3 million patients, we didn't observe depression as known side effect with this drug. So this was the first step. In terms of preclinical, we have shown that the drug has a pharmacologically is differentiated to tetrabenazine, and we have some elements that lead us to hypothesize that our drug has a better profile for the mental health. And third, in this first proof of concept trial, we didn't observe adverse event of depression or neither worsening of depression in the patient. Of course, it was a very small sample size, but half of the patients who were depressive at the entry in the trial, they remained stable and didn't have a worsening of their depression. And next, in the next trial, we will use the BDI, the Beck Depression Inventory Scale, to monitor closely the depression in the next cohort. So we hope to have the demonstration that we will have a safer drug.
So we look forward to see the data.
Yes.
I would like Bernard to make a comment, please. On it's really interesting to see that all these companies who are now sitting with us have been made use of the Enroll-HD platform. You are the PI of the Enroll-HD platform. I would like Bernard to please make some concluding comment on this use of the platform and this study.
Well,
Short comments.
Sure. It will be brief. So I feel that the collaboration of so many researchers, so many centers, so many family members on this did enable us to bring together something that is of tremendous use scientifically. For instance, that allowed us to identify genetic modifiers, that will allow us to identify environmental modifiers, hopefully at some point. And it enabled us to do a recruitment to interventional studies in a very targeted way, plus collect in a targeted fashion. And I was explaining all about an HD Clarity, vital biomaterials for important questions that would support drug development as well as the deeper understanding of HD.
Thanks. So I hand over to our major chair, Anne.
So I'm very sorry we can't continue that discussion for longer. It was really interesting. There are a lot of questions in the chat, so we ask the speakers to kindly answer those on the platform, if you wouldn't mind. We have a break now until 18:10 Central European time, and we'll start at 18:10 sharp, when we'll have the final keynote session. So please don't forget to click on that session to start it, and enjoy your break.